Domainex is a multi-award winning, fully integrated provider of drug discovery services. So we're a contract research organisation, or CRO. I think the thing that makes Domainex really special is that we work in a very collaborative way with our clients, and that's often why they choose us. We provide a lot of intellectual input and recommendations and guidance, and that's why they come back to us time and time again.
As a biology department, we're experts in target enablement. So when you've got a pill that's gone to marketplace, 15 years before then, somebody's worked heavily on that molecular target, and we've got the experts to make that, produce that molecular target, and also find hits that maybe affect the activity of that target. And those are the early compounds, those early drugs, and that enables the process to progress through to sort of 10, 15 years later, you end up with a drug to market.
Within Domainex, we've got a very well-equipped biophysics lab. This includes 3-wave delta systems and also a PEAQ-ITC instrument. The wave delta system allows us to measure the binding kinetics of interactions in real time. So this includes the association between two, for example, a small molecule binding to a target protein. The recognition between that is the association, which can get measured in real time, and also how strong that interaction is, which is measured by the dissociation. And the association and dissociation of that interaction allows us to measure the binding affinity of that binding partner. The reason we like GCI technology is that it has a number of advantages over other techniques. It's very sensitive, so we can use it for fragment screening. It can measure faster dissociation rates than other techniques. It has a disposable microfluidics, so that if there's clogging of microfluidics, then that doesn't cause any instrument issues. It's the only instrument where you can just use the dissociation of the molecular interaction to measure the affinity. That has the advantage of being much more efficient and faster. GCI also has the wave rapid method, which allows you to measure the affinity and the kinetics of an interaction just using the dissociation. This makes the experiment much more efficient, so you can save time with each experiment.
We were able to obtain really high quality data for the client, and with the wave rapid technology we had a really fast turnaround of data. The best example of how we've helped a specific client is Vespa Bio. So they came to us late spring 2021, and they wanted us to initially do some relatively straightforward binding experiments just to demonstrate target engagement, again measure the binding kinetics, binding affinity, and that supported their project so that they were able to incorporate this screen as their primary screen for their program.
We have a PEAQ-ITC system as well from Malvern Panalytical. That allows us to measure binding interactions in solution. The PEAQ-ITC instrument measures the heat of binding from molecular interactions, is extremely sensitive, and the information that we get from the heat of binding allows us to measure the binding affinity, the thermodynamics of binding, and also the stoichiometry of binding. So we can see whether there's one small molecule binds to one protein target.
We really enjoy working with Malvern Panalytical because they share our spirit of collaboration and partnership.